Welcome everyone. We are now in week 11, which deals with protein macromolecular interaction 2. And here the in the last session also, like in week 10, the module 10, we also discussed about the protein macromolecules interaction of type 1, which included the carbohydrate interaction, protein carbohydrate interaction as a ligand. However, in this module, we will discuss about the protein-protein interaction, or in short, we can term this as a protein-protein interaction or PPIs. Okay, so this is a very important topic with respect to the vaccine development, then drug development, where the peptides, where the short chain peptides can be used as a ligand, or these topics are essential to understand the cell biology in which the signal transduction mechanisms are going on in our body, and then all the then if to understand the protein folding, this topic is very much essential. Okay, so I welcome you all again in week 11. Myself, Karan Kumar, PMRF at IIT Guwahati, and now we will discuss about the topics related to the protein macromolecule interaction. So as all the lectures of module 11 have been already uploaded in the PMRF portal, and I think and I hope you all have gone through this. So the topics that you might have covered are the protein-protein interaction, as the title suggests. This is also termed as the PPIs. Now you might have studied the types and properties of PP interfaces, then binding affinity of these interactions, hotspots, the terminology you might have come across, then protein-protein binding domains, then regulation of these protein-protein interaction, and about the method of detection, how you can detect those protein-protein interaction using different experimental techniques such as yeast to hybrid system and other techniques, other biochemical methods. Now you might have studied the PPI specifics and the diseases associated with them if they are not regulated in very good manner, or as they should be as they should be interacted. If they are not in that manner, then what are the diseases that occurs due to them? Then peptides as therapeutics you might have come across, how you can use the peptides as a therapeutic molecules in your body to treat the disease or to like prevent a disease to happen or to detect. Then cellular uptake mechanism, you might have studied how this peptides and protein-protein interaction are essential to deliver the drug inside the cell, and how these cellular uptake mechanisms happen inside the organism or like biological system. Then you might have studied the protein folding using the molecules which is known as the heats of protein, HSP, and the elements that are very much important for the protein folding. Then you might have studied about the sapronin and chaperones. Then protein nanoparticle interaction, their application, method of detection, and impacts on the biological system. Protein nanoparticle interaction is a kind of very well-known field of like in this area many research are going on. Researchers are working on to deliver how the how to utilize gold nanoparticles, silver nanoparticle, and other metallic nanoparticles to deliver the proteins or peptides efficiently to the cells where it is needed, or you can also term that as a targeted therapeutics, targeted drug delivery. So these nanoparticles are used as a delivery vehicle of drugs or protein or DNA molecules, which can be utilized as a therapeutics. Then the important keyword that you might have come across as a hotspot, which are the essential amino acid where the two protein molecules interact. Then interfacial water that occurs between the two subunits when they are being interacted, the crystal water being fixed there, and then they play a major role in interacting that in protein-protein interaction. Then geometrical complementarity, SRC homology, you might have come across. Then phosphotyrosine, then peptide therapeutics, as I already mentioned here in protein, like protein nanoparticle interaction and the peptides as therapeutics. These peptide therapeutics can be like you might have come across. Then peptidomimetic protein motifs such as gene swinger motif and other motifs that can be used as the interactive molecules, and databases for these interact in like peptide interaction network. Then you might have studied about the angiotensin converting enzyme AEC, which are very much essential while regulating the blood pressure inside the cell, and these play an important role in various diseases related to the cancer and other like blood pressure related diseases. Then cell penetrating peptides and HSP, which is nothing but the heats of proteins, and these help in the protein folding and other regulatory kind of activity in the organism. Then chaperones and chaperonins, these play important role in protein folding and guiding the misfolded protein and makes them the properly folded. Then SPR, these are the techniques to identify the protein ligand interaction, which is nothing but the surface plasmon resonance sensing. This is terminology which is related to the SPR only. Then cytotoxicity, corona. Corona is nothing but how the peptides and other functionalization molecule can be incorporated on the surface of the on the surface of nanoparticles such as gold nanoparticle, silver nanoparticles, and other metal nanoparticles, which act as a delivery vehicle. Coming to the contents of the PPT, as always, we will first practice the questions, which is on the current module number 11. Then we will discuss the important topics and we will take also few doubts from you if you have any. Then we will discuss the scope of this module with research perspective. Ma'am has already discussed in much more details in her lecture videos in this module. I will also refresh those topics and also will tell you what are the current research going on this area related to this module. Now coming to the questions related to this module. The first question for your this week discussion is which among the following is or are correct about the protein-protein interfaces? So it is related to the PPI, protein-protein interfaces. Now, your statements are the protein-protein interface is 50% polar, approximately 50% polar. Then statement 2 is protein polar residues in the interface are usually charged. Then protein-protein interface is richer in aromatic residues, and protein-protein interfaces exhibit both shape and electrostatic complementarity. Your options are only 1, 2, and 3 are correct. Option B, 2, 3, and 4th only are correct. Option C, 1, 3, and 4 only. Option D, 4th only. Any guess on this? Is anyone available? Yes, Sarna.
The option 4 is correct.
Option 4
Yes.
means you are talking about the statement 4 is correct, right? Or option 4, fourth only.
No, I'm talking about four. On this fourth one is correct. Rest,
Yes.
I am not sure.
Okay. So let's recall the protein-protein interfaces. So ma'am had discussed about in her week 1 lecture, in lecture number 56, I guess. This spans from 56 to 60, right? I guess 56, yeah, somewhere in between that. So in her first lecture of this module, she discussed about that protein-protein interfaces are 50% polar. Why these are 50% polar? Because let's assume when this because the protein normally hydrophobic cores, like hydrophobic pockets are buried deep inside the protein, deep inside the protein structure, right? So that usually termed like on the surface you will have the polar amino acid residue or hydrophilic amino acid residue, so that the water molecule can surround themselves, right? So in that case, you can assume that it might be the true statement that it will be a 50% polar, because on the surface you will have the hydrophilic amino acid residues, right? Because the protein is majorly surrounded by the aqueous solution. So the statement 1 is correct, right? Statement 1 is correct, and you are saying that statement 4 is also correct. Now coming to the other two statements, polar residues in the interfaces are usually charged. Now, if let's assume if they are charged, then it may create some kind of problem while they are interacting. So when the two proteins are interacting, when there are protein-protein interfaces, then at the interface, if they are charged molecule, then uh, there might be a problem of the charge in repulsion might be a issue there. So normally when in the protein there are polar amino acid residue, and these polar amino acid residues are neutral. So statement 2 is wrong. So if statement 2 is wrong, then you have 1 and 4 correct. So 1 and 4 correct. These are happening in only B and C option, right? So now we know that option 3 is also correct. Like option 1 and option 4 is correct. Then option C here is correct, right? Then only possible solution will be C. So 2 is incorrect. The protein-protein interface is 50% polar. This is correct statement. Now option 4 is also correct. Option 2 is this statement 2 is incorrect. So only probable answer will be C, where the 1 and 4 both are correct, right? So option C is correct here. And it is true statement that the protein-protein interfaces are richer in aromatic amino acid residues. Okay. Is it clear? Is it clear, Sonu?
Yes, sir.
Okay. Coming to the next question. Your next question is related to the antibodies, which will be these topics are important in respect to your immunology also. So in immunology is also you will study this topic about the monoclonal antibodies. So the topic is more related to the therapeutic nature. So here the monoclonal antibodies act as a modulator for targeting the large PPI interfaces they are used in. Option your statements are the screening of recombinants, treatment of many cancers, and diagnostic kits. Options are 1, 2, and 3 are correct. Option B, 1 and 2 only are correct. So option C,
Sir,
2 and
option
3
C.
correct. Option C. Option C means 2 and 3 are only correct. So why do you think that this is not related, like these are not used in the screening of recombinants? Can you like here actually all three statements are correct. Why? Because monoclonal antibody, you might have studied that while you do the ELISA technique, in ELISA technique you use the monoclonal antibodies, and there are other like when you are screening the recombinants, they you use the monoclonal antibodies. They are used as the modulators for targeting the PPI interfaces. So here the all statements are correct. So option A is correct in this statement, in this question, right? These are used as a diagnostic kit. These are also used as a treatment for many cancer, and these are the regulator for modulator for screening of recombinant protein in protein-protein interfaces. Now coming to the next question, the physical contacts between the domains are crucial for functioning of the cellular machinery. Is it a true statement or false statement? Physical contacts between the domains. What do you understand by the term domain? What is domain? Can you specify what is domain? Then we will talk about the physical contacts about the between the domain.
The domains are the super secondary structures.
Yes, domains are the super secondary structure. Very good. And what are their properties?
Right.
Domains are yes, yes.
They can act as they play an important role in the enzyme. They form important supplement portion.
Yes, they form. They play a major role in like substrate binding. They play a major role in like uh, antigen recognition. Then they play a major role in singular transduction. Without if the two domains are not being connected in case of like monoclonal antibody recognition, like when antibody and antigens are interacting, then two domains should interact. Then only it will lead to a signal transduction or the molecular recognition, or somewhere if two domains, like if the hemoglobin is made up of the four subunits, right? If the all four domains are not interacting in efficient manner, the functional the cellular machinery will not function as efficiently it will operate, right? In the similar cases of the enzyme substrate interaction also, if two domains, like let us say that enzyme binding site is there, if enzyme binding site is not occupied with the substrate and the two domains does not interact, in that case the cellular machinery will not function, right? The reaction will not happen. So in that case there should be a physical contact between the domains, because this is very much crucial for functioning of the cellular machinery, and thereby it is a true statement, right? Understood? Did you understand?
So
Okay. Coming to the next question, stapled peptides. I hope you remember what are the stapled peptides. So stapled peptides binds to the estrogen receptor using the hydrocarbon link to stabilize them in a form of an which kind of like structural form of the protein. Is it a beta sheet or random coil or turn or alpha helix? Can you recall what are the stapled peptides? Yes. And what are the role of estrogen receptors? Any idea? Any idea? Okay, so estrogen receptors play an important role in regulating the estrogen hormone, and these receptors also play important role for like in many cancer cells. Like these are the release of estrogen hormones play a major role in our body for many functional many functional things that are being played by the estrogen hormone. Okay, so what are the staple peptides? Staple peptides are a short term short peptide chain which might have the 4 to 5 amino acid residue, and these are stacked by uh, like these are stacked by some functional group. Okay, so these are the short peptide chain, and they can form a different structure based on the amino acid present in their like sequence. So they can form a random coil, they can form a alpha helix, they can form a turn depending on the amino acids present in their sequence. Okay, but when the staple peptides bind to a estrogen receptor using a hydrocarbon link, are stabilized in the form of a alpha helix. Here, ma'am has given she showed one case where the in the estrogen receptor in the binding site where the cavity was lying. So these staple peptides formed a alpha helical chain that used to stabilize this estrogen receptor using a hydrocarbon link. And the answer here for the correct answer for this question is alpha helix. Okay, this was a factual question that you need to remember. Now the next question is related to antibodies. So these are related to the like structure of a antibody, human antibody. So the human antibody unit consists of dash polypeptide chains where both heavy chains and light chains are connected through dash bonds. So your options are human antibody consists of a 4 polypeptide chains and these are connected through the option B, 4 polypeptide chain and these are connected through the disulfide bond. Very good. Yes. And the structure is Y-shaped, right? These are connected something like this. These are the 4 polypeptide chain, Y-shaped. And they are connected through disulfide bridge. Okay. So this is the structure of human antibody. Okay. And option B is correct in this statement. Now coming to next question, which among the following heat shock proteins is considered to be a chaperone? Option A HSP70, option B HSP32, option C HSP30, option D HSP60. Do you remember? Which one is the chaperone? These all 4 are the heat shock protein. And can you identify?
The option D.
Option D, HSP60. Yes, yes, very good. So option D in this like HSP60 is termed as the chaperone. And why it is called HSP60? What is the terminology means? The 60?
So it has a specific molecular weight of I think 54 to some kilodalton range.
Yes, yes. In the average, it is 60. So 54 to 68, some 65 or 68, something like that. So these are termed as these play a role as the chaperone. Okay, so very good. Now I'm coming to the next question. The antigen binding fragment (FAB) region is found by the carboxy terminal and of the both light and heavy chains of the immunoglobulin polypeptide. Is it correct or true? Correct or false? Can you recall? What are the Fab region in antibody immunoglobulin? Let us say this is the structure that I drew in last slide. Okay, so this is the structure. Which is the Fab region here?
The two heads.
Two heads. These are the Fab region. Very good. So, and these are, is it a true statement? The antigen binding fragment means Fab region is found by the carboxy terminal of the both light and heavy chains of the immunoglobulin polypeptide. Is it connected to the both light and heavy chain?
Yes, sir, it's connected to the light and heavy chain B.
No, no. Here, this is the light chain. These two are the light chain. These are the heavy chain. So is it connected to the heavy chain? It is only related to the light chain, right? Fab region is only present in the light chain, not in heavy chain. This is not connected to the heavy chain. The Fab region. This is the only Fab region here. So the statement is false because Fab region is formed by the light chain. It is connected to the two light chains, right? The two light chains. It is not connected to the heavy chain. So this statement is false. Now next question is SH2 and SH3. What are SH2 and SH3? Can you recall? SH2 and SH3 domains are typically found in protein that are involved in growth factor signaling. Is it true or false? What are SH2 and SH3 domains and where they are found? Any idea? So let's go to the lecture where ma'am has discussed about this. See, in the lecture number 52, she talked about the domains that bind to the sequence of phosphotyrosine. Normally, these are the SRC homology. The SH2, the expansion of SH2 is the SRC homology, and SRC is nothing but these are the proteins that are termed as the it is. So SRC homology domain is one of the two small proteins domains found in the SRC oncoprotein. Okay, and these are the non-receptor tyrosine kinase. Normally, these are related to the embryonic development and cell growth, regulation of embryonic development and cell growth, and they also participate in the progression of these are the oncogenes. So these will be related to the cancer, right? So here is the SH2 homology, like SH2 domain. So these bind to the phosphotyrosine and these are normally found in the growth hormone. They are, see, I will show you the, yeah, this slide I was talking about here. So the SH2 and SH3 domains both will be found in these are domain of the cytoplasmic tyrosine kinase PTK and these are related in the growth factor signaling hormone. These will attach to the growth factor signaling hormones. Okay. So now the this will be involved in the growth factor signaling. Okay. So is this a true statement or false? This is a true statement, right? Both SS2 and SS3 are related to the SRC gene. And these are nothing but SRC oncoproteins, which are essential part of the growth factor related proteins, and this will be related in the growth factor signaling, which are the part of the phosphotyrosine protein. Now coming to the next question, saponin primarily bind to dash amino acid residue in proteins. Where does it bind? Is it binding to the charged amino acid residue or aromatic amino acid residues or hydrophobic or hydrophilic? Where does the saponins bind? Do you remember? Any idea? Actually, chaperones, they are primarily bound to the hydrophobic amino acid residues in the protein sequence, like in the proteins, because when they bound to the hydrophobic, there is a hydrophobic interaction, and then they help the proteins to fold in proper manner. These are the protein folding machineries. Okay. So option C is correct in this question. Now coming to the next question, which among the following about the molecular chaperones is incorrect? You need to find the incorrect statement. It stabilizes the unstable structure in by binding. It alerts the protein structure. It alters the protein structure. It repairs the misfolded or aggregated proteins, or it assists in de novo protein folding. Which one is the incorrect statement? Option B. Yes, very good. This is an incorrect statement. It does not alter the protein structure. Okay. However, all three statements are correct. Coming to the next question. The amyloids, which also you can term them, you might have heard about the beta amyloid protein. Okay. So amyloids are comprised of cell nuclei, glycoproteins, fibrillar deposits, or metalloconjugates. Where do you find the beta amyloid proteins?
When there is a misfolding of protein, there is an aggregation of all these amyloid structures.
Okay.
Sir, should it be option C?
Option C. You are talking about the option C, which is the fibrillar deposits. So are you sure about it, or you are just guessing?
No, sir, I'm guessing it.
Okay. Actually, this is the correct answer. Fibrillar deposits. Why? Because when the protein is not being folded properly, they will form as a seed kind of structure, or the resid, or kind of a fibril kind of structure. And when these fibril kind of structure got aggregated, then they are termed as the amyloids. Okay. And these amyloids are the main cause of Parkinson's disease. And the form of the protein that found in the Parkinson's disease, these are the beta amyloids. Alzheimer and Parkinson's, in both of the diseases, neurodegenerative diseases, beta amyloid play a major role, and these are comprised of the fibrillar deposits. Okay, option C is correct. Now coming to the next question, domains that primarily bind sequences with phosphotyrosine moieties. Phosphotyrosine moieties such as which are PTS are SRC homology 2 SH2, SRC homology 3 SH3, or PTB. So your options are 1, 2, and 3. Option B 2 and 1 and 2 only. Option C 1 only. Option D 1 and 3 only. Where does the uh, where does the primary sequence primarily bind the sequence? What with the phosphotyrosine? Any guess? Just now I showed one structure related to this, like phosphotyrosine residues, like in where the SH2 and SH3, all the domains were visualized. Any guess? Where does the phosphotyrosine moieties bind primarily? In which domain? Let us go to the slide again. Okay, so this is a protein where it has the SH3, SH2 domain, then kinesin N domain and kinesin C domain. This is called the SH1. Okay, now these all are linked through a linker and it has a tail. Okay, so when you see, when you go to the previous slides here, the primary sequence bind to the, let us see here, SRC homology. Phosphotyrosine, like PTB, phosphotyrosine binding domains interact with the sequence containing phosphotyrosine group. It is clearly mentioned here. And also, if you do not like remember this, you can also analyze that by the structure. In SH3 and SH2 domain, the kinase domain is not present. However, in SH1, where the PTB binds, this is the PTB domain, where the kinase N domain is present and kinase C domain is present. These are this is where the kinesin tyrosine PTB will bind, like this tyrosine, phosphotyrosine will bind. Okay, so PTB, the correct answer here, PTB binds. Then coming to the next question, like next here, in SH2 also, it binds with the phosphotyrosine residues in the target protein. Okay, the peptide usually has the beta conformation. Now, when you are coming to the SS3, same to the SS3. Let us come to the SS3. Yeah, okay. In SS3, which is the proline rich region, it is not mentioned here that it will bind to the phosphotyrosine or not, phosphotyrosine sequence or not. So here in question number 12, this will only bind to the SRC homology 2 and PTV. Okay, so your correct option in this question was option D. It binds to the both SS2 domain and PTV domain, but it does not bind to the SS3 domain. Okay. Now coming to the next question. Choose the correct statement about the database of interacting proteins. Database of interacting proteins, DIP. Your options are: the secondary database combines the experimentally inferred data taken from primary database with computational prediction of molecular interaction. Option B, the predictive database combines the experimentally inferred data taken from the primary database with computational prediction of molecular interaction. The predictive database combines the experimentally inferred data taken from secondary database with computational prediction of molecular interaction. Or option D, the secondary database combines the computational prediction data taken from predictive database with experimentally inferred data of molecular interaction. So Can you recall this about the database of interacting protein?
Explain this.
Yes.
So can you please explain this?
Okay. So here the database of interacting proteins. This ma'am discussed about this in the last lecture, I guess, in the last or second last lecture. Okay. These database of interacting proteins are nothing but when you have a PPI network, protein-protein interactome. Like why this is essential? When there are different database such as primary database, then secondary database, then computationally derived database. So in that, and computationally derived database are also known as the primary database, secondary database, and predictive database, which are kind of tertiary database. Normally, the predictive databases, the first primary database contains all the protein-protein interaction database which are experimentally derived. So when they are reported, then the people curates from the literature and then they incorporate into the primary database. Okay. So normally primary database contains the experimentally identified the protein-protein interactions. However, in secondary database, they use the experimentally experimental techniques and somewhere in the and they use the analogical techniques and also some of different techniques where if A is interacting with B and B is interacting with C, then they infer that A might be interacting with C or it might not. So these are analogy based or secondary kind of they also contains the experimental database but with analogy. So while in the predictive database, these are extended, and because we know there are multiple proteins, there are many proteins and number of proteins available in the human organ and different organisms. So if we try to if we try to study through the only experimental techniques, then it won't be possible because there are hundreds or there are millions of proteins available, millions or billions of protein in astronomical units. So studying that experimentally will be very exhaustive. So there are different molecular dynamics techniques and other computational techniques which can predict the protein-protein interaction in computational ways. So in the predictive database, normally we use the experimentally inferred data and primary database, and then we put that into the predictive database. So these are the hybrid database where we take the inference from the experimentally derived and we use the computational techniques with the molecular interaction. So in this, if we check now with this inference, if we check that statement, the secondary database combine the experimentally inferred data taken to the primary database with computational prediction of molecular interaction. This statement is incorrect actually. Now, the predictive database combined the experimentally inferred data taken from the primary database with computational prediction of molecular interaction. Yes, we use computational techniques in predictive database. So the statement B is correct here. However, if we see the predictive database combined the experimentally inferred data taken from the secondary database of computational prediction of molecular interaction, this is not a correct statement. So option B is correct here. The predictive database combine the experimentally inferred data taken from primary database with computational prediction of molecular interaction. We do not take from the secondary database. We take from the primary database. The option D, if we say the secondary database combined the experimental prediction, computational prediction. Here it is wrongly interpreted because we take the experimentally inferred database. So the correct statement here is option B. Am I clear?
Yeah.
Okay. Now coming to the next question. Which statement is or are true about the limb domain? Okay. Limb domain. It contains the tandem cysteine rich ZN2 plus finger motif. It binds to PDZ domain. It folds into a zigzag triple stranded beta sheet, or it plays a role in cytoskeleton organization. So which statement is true? We need to identify the true statement. Options are all four statements are correct, or 1, 2, and 4 only are correct. Option C, second, third, and fourth are correct. Option D, first, second, and third are correct. So can you identify the correct statements in this question? Do you remember about the limb domain? If you remember about the limb domain, it does contain a cysteine-rich ZN2 plus finger motif, and it does bind with the PDZ domains. But it does not fold into the triple stranded. However, it have the 7 beta strands, and these are connected by the random coils. So statement 3 is incorrect, and it plays an important role in cytoskeleton organization. This is also a correct statement. So here, if you see the first, second, and fourth are correct. So option B is correct in this question. Now coming to this, if we see about the limb domain, this you will find in lecture number 53, I guess. Sit here in this limb domain. Let me check. Maybe in 454. 54 is related to the sapronins. Will not be there, I guess, in 52. Even. Where is limb domain? Yeah. So here, these are found in like the same thing. Like WW, there are different domain, WW domains. So these are the structural module involved in the protein-protein interaction through recognition of protein-rich peptides motif, and mediates the regulatory protein complex in various signaling network. Now the PDZ domains, the structural domain of 80 to 90 amino acids, and this recognize the carboxylic terminal of tripeptides motifs. Other PDZ domains or limb domains. So limb domains recognize the PDZ that binds to the PDZ domains. This is a correct statement. Now binds via a short peptide sequence which has the C-terminal hydrophobic residues. PDZ domains, some proteins with PDZ domains are identified as scaffolding proteins, and involved in ion receptor assembling the receptor enzyme complex formation. Now, about the limb domain, it is a conserved cysteine-rich sequence found in eukaryotic proteins, and it contains a tandem cysteine-rich ZN2 plus finger motif, binds to a PDZ domain, specific transcription factor, and other limb domains. This is a divergent amino acid sequence that facilitates the formation of a stable protein core and tertiary fold, then possibly high affinity binding to the wide variety of protein targets due to the double zinc finger structure. It has a double zinc finger structure, and it plays a important role in cytoskeletal organization, organ development, regulation of gene transcription. So here it does not fold in the zigzag triple stranded beta sheet, right? You can see the structure here. You can see the structure here. It has a kind of like double zinc finger structure. Okay, and it does have the role in cytoskeleton organ. It does bind to the PDZ domain, and it contains a tandem cysteine-rich ZN2 plus finger motif. So option first, second, and fourth are correct. Now coming to the next question, there are dash number of protein considered as dash connected nodes with interact with dash proteins that are known as the dash proteins. Identified the correct answer in this. Option here is like A is limited, B is highly connected, C is many, and D is hub. So option B is limited, poorly, small, and hub. Option three, statement 3 is large, poorly, small, and non-hub. And sorry, large, highly, many, or non-hub. So you have two different you have two concepts here about the hub and non-hub. Can you recall what are hub and non-hub proteins? Then you will be.
So yes. Hub proteins are those which are having maximum connections with other proteins in the pathway.
Yes.
Means and what basically are non-hub these proteins? Are the proteins which are targeted for any treatment.
Yes, these are because these are interacting with maximum number of proteins in the protein network, right? And these are the major node. These are the major node which are highly connected, right? So what could be the correct statements? Can you find out based on this understanding? Can you find out the two statements? Okay, let me help you in this. Right. Let's put all the four options in this statement, and then we will check whether this is a correct or not. So option A, there are limited number of proteins considered as highly connected, interact with many, are known as the hub proteins. Does it feels it is a correct statement? Limited number of proteins considered as a highly connected nodes interact with many proteins are known as the hub proteins. Is it a correct statement? Only few proteins in a protein network are called hub protein, right? So this feels like a correct statement because only a few number of proteins which are highly connected and that interacts with many proteins, those are termed as the hub proteins. So this seems correct statement, right? Is it?
Yes, sir, it sounds correct.
Yes. So option A is statement A is feels like correct statement. Now coming to the next statement, limited, poorly, then small,
This one is wrong.
sorry, yeah, small. What happens? Small and hub. Is it true? No, right? This is a false statement. This is wrong. Now coming to the next. Now coming to the next one. There are large number of proteins that considered as a poorly connected nodes interact with a small number of proteins and known as the non-hub. Is it true?
This also sounds correct.
Yes, very good. Because in a protein network, if there are many number of proteins and there are few, there will be only few proteins that will be connected to the all. However, there will be a large number of proteins which will have the small network. Okay, and that will be termed as the non-hub. So statement 3 is also correct. Now check. Both statements are only present in option A. So option A is correct here. Right. Let's check the fourth statement also. Large number of protein, highly connected. Large number of protein, highly connected. Many interact with many proteins are known as the non-hub. Is it a true statement? No, right? This is a false. So only 1 and 3 are correct. So option A is correct in this question. Now coming to the next one, the protein corona formation. Protein corona formation on the nanoparticle surface depends on which all factors? Option A, size of the nanoparticle. Option B, shape of the nanoparticle. Option statement 3, the media composition. Your options
The
are
option D.
Option D. Media composition. Does it really depends on that?
The material with which the nanoparticle is going to be made. Like if it is made of gold, it should have different properties.
Yes, very good. So option D is correct here. This will depends on the all three factors. It will depend on the size of the nanoparticle. It will depend on the shape of the nanoparticle, what kind of shape it has, and it will also depend on the media composition. That means the corona formation means the functionalization of the surface of nanoparticle. So if it has a different shape, then it will impact on the corona formation. Then if it has a different size, then also it will impact on the corona formation. And media composition means the composition of nanoparticle, the concentration of nanoparticle and concentration of proteins. Concentration of proteins or the functionalization molecule, both the composition will impact on the formation of corona. So all three statements are correct. Now question number 17, which you need to identify which of the following is a true statement about yeast to hybrid system, Y2H. Y2H screening. So your options are the yeast to hybrid system uses transcription of a reporter gene driven by GAL4 transcription factor to monitor whether or not two proteins are interacting. The first thing first, used to hybrid system is used for the protein-protein interaction, right? These are experimental techniques that are used to identify the protein-protein interaction. So you need to filter out the situation where the protein-protein interactions are there. So I think all four are talking about the protein-protein interaction only. So let's read out the all four statement. the DNA binding domain chimeric protein will not bind upstream of the reporter gene. If the activation domain chimeric protein interacts with the DNA binding chimeric protein, the reporter gene will be transcribed. Or the disadvantage of this method are the only pairwise interaction are tested, as well as the highly false positive rate. So here it is given, like the diagram for the yeast to habit system is given here. So as you can see here, the bait is attached with the DNA binding domain and prey is attached to the AD. When prey and baits are interacting, then only the reporter gene will be transcribed. Okay, otherwise transcription will not happen. If these all the subunits will interact, then only the reporter gene will be transcribed in the nucleus. Otherwise, it will not report. So, and if this is transcribing, that means the two proteins are interacting. Right? So now can you identify which statement will be correct? So does it use the transcription of a reporter gene by GAL4 transcription factor? Is there any GAL4 transcription factor involved here? No, right? It involves the bait and prey, two kind of proteins, and therefore this is a false statement. Now talking about the option B, the DNA binding domain chimeric protein will not bind to upstream of reporter gene. Where is the upstream and downstream of the reporter gene? Which region is it is? 1 and 2. What is 1? Is it a upstream or downstream?
So 1 is the upstream.
1 is the upstream. Yes. And second is the downstream.
Downstream.
Right. So where is the reporter gene bind? The DNA binding domain chimeric protein will not bind to the upstream of the reporter gene. Will it bind to the upstream? US? No, right? It is binding to the bait. So this statement feels like it is a true statement, right? Now coming to the next, let's keep this B as a like unidentified one. We are not so sure about it. Now coming to the option C, if the activation domain chimeric protein interacts with the DNA binding domain chimeric protein, the reporter gene will be transcribed. Is it a true statement? If the activation domain chimeric protein interacts with the DNA binding domain, chimeric will it bind to the DNA binding domain? Or will it bind to the prey or bait? It will bind to the bait, right? So this statement is also incorrect. If the activation domain means AD. If the activation domain chimeric protein interacts with the DNA binding domain chimeric protein, the reporter gene will be transcribed. No, the prey will interact with the bait. So this statement is also false. Now disadvantage of these methods are only pairwise interactions are tested as well as high false positive rate. No, this is also not a correct statement because it has a very less false positive rate and this is a highly precise technique to study the protein-protein interaction. So this statement is also false. So among these all four, B statement feels to be right. So correct statement about this is the DNA binding domain chimeric protein will not bind to the upstream of the reporter gene. However, it will bind to the prey will bind to the bait and the transcription will happen. Okay. So option B is correct in this question. Now coming to the next question, which is related to the cell penetrating peptides. Cell penetrating peptides are the peptides that are able to penetrate biological membrane and trace transport of the bioactive molecule into the cells. Option A, short. These are the short peptides and facilitate the transport. These are the long peptides and facilitate the transport. These are the short peptides and inhibit the transport. These are the short peptides and these are the long peptides and inhibit the transport. Which is the correct statement? It is given here, the CPP. The structure of CPP is given here. So can you identify? Is it a short peptide or long peptide? First tell me that thing.
Short peptide.
Short peptide, very good. And does it facilitate or inhibit the transport of the bioactive molecules? Facilitate, because these have the interaction with the membrane proteins, and by which these are transported directly through the membrane, and if that is attached to a bioactive molecule, the bioactive molecule will also go inside the cell membrane, and thereby it facilitates the transport of bioactive molecules. So option A is correct here. These are the short peptides that facilitate the transport of bioactive molecules into the cells. Now coming to the last question of this module. So identify the amino acid residues with the number of angiotensin 1 shown below, which are cut off by AEC enzyme to produce the angiotensin 2. Can you tell me what is the role of angiotensin 1 and angiotensin 2? Can you tell me? What are the role of angiotensin 1 and angiotensin 2 and where it plays a role, major role? AAC is also known as the angiotensin converting enzyme. AAC is also known as the angiotensin converting enzyme, and it converts angiotensin 1 to angiotensin 2, which play a major role in blood pressure regulation. When the blood pressure is low, that means what? Can you tell me, when blood pressure is low, will the blood flow is higher or blood flow is lower?
The lower. If it's low blood
pressure. Low blood pressure. When the blood pressure is low, the blood flow is higher or lower?
Sir, lower.
No, you are wrongly interpreting it. When the blood pressure is low means the the tubes which carry these channels which carry the blood are dilated. Like these are the more broadened, and then the blood flow will be higher. The blood flow is not obstructed. That is why the blood pressure is low. Okay, so in low blood pressure case, the blood flow will be higher. So to obstruct that, angiotensin 1, what does it do? It contracts these vesicles, and that is why the blood flow retains in there, and that it creates the pressure in this vessel, in these vesicles, and then it creates, it normalize the blood pressure by obstructing the blood flow. It increases the blood pressure, and the low blood pressure now comes to the moderate state. Okay, so the role of angiotensin 1 or angiotensin 2 is to regulate the blood pressure, that we know now. Now, what does the angiotensin converting enzyme do? It cleaves the structure at 9 and 10 amino acid residues, so that this fragment is now released, and this angiotensin 1 is the complete molecule. When these two fragments, which contains the amino acid 9 and amino acid 10, are released from this structure, now this is converting into angiotensin 2. So option B is correct in this statement. This is the histidine amino acid residue and this is a leucine amino acid residues. So leucine 10, histidine 9 is being chopped off at the carboxylic acid terminal, and then this angiotensin 1 is being converted to the angiotensin 2 by AEC. Okay, so option B is correct in this question. So by this, we are done with the assignment practice for week 11. If you have any doubt, you can ask your questions, your queries. And now coming to the discussion on important topics. The important topics in this with respect to the research perspective is when you study the protein-protein interaction. Protein-protein interaction are normally studied by the yeast to habit system. But as I already mentioned in somewhere in the slide, that these experimental techniques will not be much efficient to study all protein-protein interaction that all interaction that are in the human or a single organism. So the computational techniques are much more important to study the protein-protein interactions. So these are the very much relevant area in research. The computational technique to study the protein-protein interaction and to identify the protein interaction network. Because if we find out the hub protein, if we identify the hub protein and we knock out that protein, and if we inhibit that particular protein and then we check whether the phenotype is changing or that particular disease is being cured. So in that kind of scenario, we will be able to identify a more potent drug to treat that particular disease. So to identify the hub protein, we need to have the information of interactions of the different protein and how they are being how these are being interacting. Is it inhibiting the enzyme or is it promoting that particular if the regulation of this and this particular protein is upregulating upregulating the expression of this protein, or it is downregulating, or it is not interacting at all. So these kind of study are very much important to identify the drug target. Okay, the another important aspect of protein-protein interaction is related to the here to identify the domains, like which all domains, like if two proteins are interacting, then to identify which all amino acids are important for interaction of these two proteins. If we identify this via molecular dynamics analysis or molecular docking analysis, then that will be a very much fruitful study to identify a drug target. Why? Because if we modify that particular amino acid, and then these two protein will not interact, and thereby it will not do the signal transduction, and we can inhibit the cancer progression. Okay, the same concept can be altered, or it can be like it can be checked in the different way. If we want to express certain kind of regulation, if we want to overexpress a gene, and for that the interaction should happen, in that case we need to check we need to check particular like that we need to check the particular domain where the drug is binding to, or the antigen is binding to that antibody, that particular domain, if we identify, then that will help us to design the drug target, right? To overexpress that particular hormone, which can limit the progression of that particular disease, right? So thereby, using the protein-protein interaction techniques, you to identify the domains, interacting domains, we can know which kind of drug targets we can implement. So these are the two research domains which are being regularly practiced by the researchers in scientific community to identify the drug target, to understand the disease properly, and to understand the mechanism of transduction. So with that perspective, this particular field is very much important in research perspective. Okay, I hope this is clear to you. So are there any doubt? Are there any doubt?
No,
let's
sir.
say for
Okay. Thank you, Sanva. So thank you for joining. And this is our second last module. We have only one module left, which is week 12, and that the lectures might have been uploaded already in the NPTEL portal. By that, we will have the last interactive session of this course, and then that will be all for this course, Fundamental of Protein Chemistry. So if you have any feedback, you can give that in the feedback forum, or you can ask your question in the forum of this module of this course, and also you can guide your questions in my email box if you have any. Thank you for joining. That will be all for this interaction. Yes. Is there any questions or no? No, I could not hear you properly.
Change in the ligand site is the presence of a background concentration that can vary over time.
Hello. I'm not able to hear you properly.
Sir, can you listen it?
Yes, yes.
I said thank you, sir.
Okay. Thank you so much. There was some background noise, I guess. That is why. Okay. Thank you so much. So that will be all. And I hope you will join in the last session. So thank you.